Hello, P-Chemists. Let's get into your questions. Before we start your questions, I want to make a quick announcement. Things are piling up, and I realize that the presentation is due on Wednesday already. Please default to going ahead for that. But if you need more time and you feel like things are piling up too much for you, and if you would prefer to write rather than present, I want to give you the flexibility for that. And so I will allow you to, instead of the presentation, write a 1,500-word paper due with the final. That will give you another week to do it. Same format, same idea. I would like to see 2 peer-reviewed papers that you talk about and the same topic. I'd like to see you pick something, pick an element. The one other thing to realize is I won't be able to write a test question related to this like I was thinking I would be able to for the presentations. With all that said, if you can do the presentation, I recommend going ahead and doing it. If you need more time or want to write rather than speak, go ahead and go for this. You have my permission to do whatever, and if you need any flexibility for any of the other assignments, definitely let me know that as well. But to get to the questions that you have, which also give me ideas for test questions that I might write, we got a bunch of good questions, and these are kind of complex questions because we're in complex chapters in a complex phase of natural history, right? And so my answers may not be completely adequate to the quality of your questions. They might not be very good or fulfilling, but I'll do the best I can, and I'll give you examples of some of the ideas that these give me. So with all that, saying I'm a chemist and I'm talking about things that are sometimes a little bit out of my view, but these are the kind of questions that we want to ask. And so I think it's great that you're asking these questions. Let's see what I think the answers might be. Chapter 10, but let's start off with the story of the pandas losing their meat tasting gene, the umami tasting gene, which is like a terrible thing. Umami is one of my favorite things, right? So why would you lose it? Well, the umami is an incentive to eat meat because it's glutamate. There's a lot of glutamate in meat, and actually there's a chemical reason for that because there's a lot of calcium in mussels. There's a lot of glutamate to bind the calcium in mussels. Glutamate's good at calcium binding. You end up with a lot of glutamate, and you end up with a taste receptor for glutamate, which is the umami taste. That's kind of cool. But isn't it more beneficial for the organism to have food options? And that is true. But the thing is, the cost, you always have to consider the hidden cost. There's a cost to every protein you make, and even every gene you maintain in your genome. There's a cost because you have to replicate that gene, you have to make that protein, you have to put the energy into making that organization. Remember, we're always fighting over the pull of disorganization and entropy, right? So there's a cost to maintaining the gene, and if you don't need it, there's a trade-off at some point where the benefit you'd get from being able to take in this energy is outweighed by the cost of keeping something available that never takes in any energy in real life. How that cost is negotiated is a very interesting question in evolution. But the thing is, if you can't ever eat meat, why do you keep the meat gene turned on? Why do you keep investing in keeping the meat-eating genes there? So it's wasted energy to make a protein that's never used. And that's one of the reasons why you might want to... excuse me. Hopefully that didn't blast out the microphone. You have allergies. You have to say that all the time now whenever you sneeze. You have to clarify that it's allergies and it's not COVID. Or, by the way, we're safe anyways because we're doing this by teleeducation. Okay, so anyways, back to the meat tasting gene. It's the cost-benefit analysis of making proteins that you'll never use. And if you never eat meat, you don't ever use a meat tasting gene. That would make it beneficial for you to take in energy through meat. And actually, so the panda pays a price. The panda is a huge animal. It has to eat a ton of bamboo. Basically, it has to sit there and eat the bamboo all day. But it doesn't get very much energy out of the bamboo. And so the entire panda, you don't think of, if you wanted to get in a race with a bear, you'd probably pick the panda bear, right? Because the panda bear, it doesn't seem like they go very fast. You have the image, they're kind of a Pooh Bear type image, where you have the panda sitting around all day and just munching on bamboo. And there's a real energetic reflection to that. And in fact, that goes all the way through to their metabolism. To be able to manage on a more restricted diet, to be able to manage without high energy meat, they manage on low energy bamboo by turning down their metabolism. And remember that metabolism has a simple chemical physical correlate. The more metabolism you do, the more heat you radiate to the environment. That's a very simple thing that has a lot of caveats to it. But the idea is if you're comparing similar animals, the ones with lower metabolism, and I should say probably lower catabolism is what we're really talking about here, are going to be colder. And they actually did this, and here's something where they measured the energy that pandas do. And it turns out that there's a whole argument. There's bunches of arguments. Most of the arguments are more scientific, but here's where they just pointed a thermometer at different animals, and they compared that the other animals are much brighter, whereas the panda is more dim. That means that its surface temperature is lower. Okay, and they compared this. There's a lot more better graphs than this, but this is kind of a cool graph that pandas are cold. Pandas run at a lower temperature because they are processing less energy, and they are less in thermal disequilibrium with their environment. They're closer to thermal equilibrium with their environment. That's one of the ways in which they manage to eat bamboo all day. If you want to have a higher temperature, if you want to run faster, great. But you're going to have to take in more energy than just bamboo. In all the trade-offs, the pandas have done this. And there's actually a better article about this in C&E News, but I couldn't find it in a quick search. If you're interested more in that, look up C&E News and panda. So another question from Chapter 10. Talking about neurons forming paths through the brain, and parents always say your drinking age is 21 because at that age your brain stops developing, and it can mess up your development. Were my parents right or not? The answer is, I'm going to back up your parents on this. They were functionally right, although to a lesser degree, it's really a spectrum rather than a binary. You have more brain neurons being developed as the brain is growing when you're an infant, and as it's developing during adolescence, you are doing more of the brain-forming chemistry. And so that would mean that the alcohol, which is a solvent that saturates the brain and changes the way the brain works, sort of inhibits it in most areas of the brain. And so you don't want to inhibit development. And development does happen more when you're young. I think learning continues to happen, so it's why you shouldn't be drunk all the time, even if you're 83, But again, we're talking about the trade-offs of alcohol. One of the costs of alcohol is that it does impair brain development and learning. Because that is a greater risk for those who are developing their brain more, that's one of the reasons why a drinking age makes sense. Now, should it be 21 or 18? Should we be European? Should we be American? That's all where you draw the line on the spectrum. I'm not going to comment on that, except to say that there are definite connections. But also, we don't know entirely how all those connections are, which ones happen when, the detail of those connections. All I can say is this argument does actually make physical and chemical sense. But that's cost-benefit analysis might be one of the themes of my Q&A today. Here's another one about neurons, talking about the importance of calcium as a signal in neurons. And, fast neuron activity can lead to fast evolution. Cockroaches are able to avoid cockroach poison and to literally evolve. Now, would a deficiency of calcium in the diet or in general lead to slowing down evolution because of slower neurons? It's unlikely to be a big enough effect in the diet of humans. Not in humans, and by the way, you have a calcium reservoir in your bones, and when you need calcium, you actually have osteoporosis happening where you pull calcium from the bones. So the thing that low calcium would affect is not your brain, but your bones. Because the brain is so important that your body will take from the bones to give to your brain. So that means for humans on a short time scale, we're not talking about a measurable effect for calcium doing this. On the other hand, sugar will have an effect short term because sugar is your short term fuel for the brain. And that's why I suggest drinking sugared drinks before a test, because it will actually get your neurons firing faster. I think there's something to that. So the question is, calcium in general, maybe for another organism like cockroaches, well, I think it'd be an interesting experiment with cockroaches. I believe that definitely if you did it in bacteria, the kind of calcium signaling that you would do could be influenced by the calcium in the environment. Cockroaches, they don't have bones to pull from, so it'd be an interesting kind of question to see if the calcium threshold could affect neuron function. You could experiment on that. And also, when you say in general, I also think back billions of years, and I think about, okay, there's a paper that has evidence that the Cambrian explosion type of evolution, the morphological evolution of shapes of shelled creatures, happened more in environments that had more calcium as opposed to magnesium, calcium-rich environments. That's for shells. That's for body shapes. Is there also a correlate for neurons? That would be a very interesting thing to do, but it's hard enough to do for shells, which are preserved in the fossil record, as opposed to neurons, which are not. But I want to say, yes, when you're asking a question like this, it's great. Just basically look at all the different ways that you can ask that question. And I want to ask it with you. So keep asking it. If you find I think it's unlikely to cause an effect in humans for the reasons that I mentioned, but think about what experiment you would do to test it. That's the cool thing about this book, in my view. Once we get to these chapters, I'm getting to the point where I'm saying, I don't know. Why don't you test it? Or why don't you look it up? That's the great part as a teacher, when you can sit back and say, okay, I've done what I can. Now you all take these ideas and see how they work in your daily life. And to me, that's the goal of the class. Okay. So now we have 2 Chapter 11 questions to wrap up. And this is one about eusociality, which I talked about the chemical communication within eusocial species. Is it like hormone signaling? And that is exactly what it's like. You make a particular chemical shape out of a small molecule, and you use that to communicate to a receptor in other cells. The receptor in this case, in hormones, the receptors are, the communication is sent and received within one's own body. The only real difference here, and I'm speaking at a very vague chemical level, but the major difference here at the chemical level is that the signal is made from a small molecule sent from one creature and received by another creature. These are often called pheromones, and there's debate over whether pheromones are even important for humans right now. But let's, not to enter into that debate, I can say that they are important for bees, and they're very important. What's really interesting is not all bees use these, but when bees use the molecules for communication, they will use the same molecules as wasps do. So you have this really interesting case where eusocial bees, eusocial wasps will use the same molecules, even though they are separated by evolutionary divergence. This is the example of convergence. And I want to show you my favorite paper about this. This is about queen pheromones, which is the signals that go around and say, hey, you're a queen and you're a drone. You know, they send that kind of signal throughout the hive. And definitely going from one animal to another. I believe these are mostly volatile, although they might be somehow soluble. They might be secretions or something. But I believe that enough of these will get into gas phase that they can actually go around, and it would be like smelling something. That's how, by the way, when they look for human pheromones, they always are looking for the sense of smell, because that's the way that you detect complex molecules through the air. Okay, so this paper looked at 64 species of social insects, and they found at least 3 independent origins of eusociality. And these independent origins used very similar molecules despite being separated on different branches of the evolutionary tree. Here's the data, and I just show this to you to show you the 3 origins. You have wasps at the top, bees in the middle, and then you have ants at the bottom. Those are different, and there are definitely differences between the way that those do queen signaling. But look at the molecules the wasps use at the top. They're all those saturated hydrocarbons, which, by the way, are shown as red on this graph. The red boxes mean they signal with saturated hydrocarbons. Look at all those different species that are signaling with the same molecule to send the same signal. And they're sending, and they really are the same molecule. If you look up and down between the A, C, D, very different species, somehow using the same molecule to signal, hey, you're a queen, hey, you're a drone. Now, there definitely is diversity. If you look in the bees, you see a lot of colored boxes, and you see that the bees have a blue box, and they have different shaped molecules. The bees have a more diverse fundamental thing of signaling. The main thing I want to show you is there's diversity and there's convergence. But I believe when I look at this, I see all the convergence where I did not expect it. I expected the diversity like what we see in bees. I didn't expect wasps and the one kind of bees in the sea and wasps and ants to be the same. And I think that the similarity is more striking than the divergence. And I do think that biologists are brought up to sort of emphasize the divergence. It's part of being a biologist. You love all of the emphasis of the diversity of the world around us. But I think that along with the divergence, there is another strain that we can look at, which is convergence, where they split apart into different species, then the species come together to use the same chemical mechanism, even the same chemical exactly, to send the same signal, despite their divergence. It's a complex thing. This is a complex paper, but I think this is really, really cool. And it's definitely a debatable point, where you're talking about not a binary, but you're talking about a spectrum. How important is diversity? In my view, Stephen Jay Gould took the binary edge and he said the only important thing is the diversity. And I'm like, wait a second, look at this. There's a lot of convergence that's important as well. So these kind of questions are cool. One last question from chapter 11. It talked about Arrhenius equation graphs. Are the products always going to be lower in potential energy? And this is a great PChem question, so I'm glad that you're asking questions like that. Are there situations where the products can be higher in potential energy than the reactants? And the same student says there's a connection between the Arrhenius equation that we talked about and the energy rate density that we talk about. Great, see those connections. So that's what I want you to see. And maybe I can ask you a question about energy rate density that will walk you through this exact connection on the test. I don't know. But the basic question is, do we have to always write it a reaction so that the products are lower energy? And the answer is no. We can write a reaction any way we want. Now, we tend to write it in the spontaneous direction because we tend to study spontaneous reactions. Chemists study things that happen. Because of that sort of cognitive bias, we actually will tend to show you reactants going to products. Something happens, goes down in energy, and there's an activation energy up here. But realize that, those two levels can be the same. They can be up. It's just all a question of what you call your reactant and what you call your product. The only difference is that your delta G depends on how you write the reaction. So if you write the reaction with reactants lower than products, you can do that all you want. You're just going to have a positive delta G. You're going to be going up when you end at the end state. We usually write things as exergonic equations, just reactions, just because the reaction is spontaneous. You don't need to add energy in to make the reaction happen. But when you're studying something like, say, photosynthesis, you are studying an endergonic reaction. If you're studying ATP synthesis, anything synthesis, you are usually studying an endergonic reaction, and life is somehow paying for that positive delta G with the negative delta G of ATP hydrolysis, with the negative delta G of sunlight, radiant light coming in and raising the energy of electrons in the case of photosynthesis. There's all sorts of things. And so it can get pretty complicated, but when we're talking about a single reaction, we can draw it any way we want. Anything we can draw as an exergonic reaction, we can just flip it around and draw it as an endergonic reaction. And if you remember what you did with Hess's law, you essentially do this. Remember? Hess's law, you take a reaction, let's say it has a positive delta H from reactants to products. If you flip it around, products to reactants, then the delta H becomes negative. That's exactly an expression of what we're talking about here. The only extra ingredient is delta G is not just a measure of energy, it's a measure of spontaneity. It's a measure of whether something will happen. And the big PChem concept here, the reason for that is delta G is related to the S of the universe, the entropy of the universe. And because the entropy of the universe is always increasing, the delta G of anything that happens, just like the universe will expand, anything that expands and has an entropy enough that expands, the delta S will be positive, and if the delta S of the universe is positive, the delta S of the reaction is negative, the delta G. Anyways, I should write these out instead of talking these out. You get yourselves into trouble when you do that. But thanks for listening to this. I won't take up any more of your time so that you can get to work on all the work that I've assigned you. But do keep on doing it. You're seeing how all these things come together. And that's my goal. And I'm seeing it come together in the questions that you're asking. So let's keep it up. Let's move through the last couple weeks of class. And I look forward to seeing your presentations and or papers. Or, there's not an and in there. Okay.